good morning. So thank you very much for the very kind introduction. So I am very, it's my great honor to give a talk in this seminar series. So today I'd like to talk about something related to autophagy and its related pathways. Okay. So I don't think you need any more introduction. So let me just remind you what autophagy is. So autophagy is an intracellular degradation process. So when autophagy is induced, a very small membrane sac elongates and encloses part of the cytoplasm, which results in the formation of this double membrane structure, autophagosome. So then the outer membrane of autophagosome fuses with lysosomes. Then lysosomal enzymes enter into the autophagosome and degrade the engulfed materials, and resulting amino acids are delivered back to the cytoplasm and used for many different purposes. So autophagy is a kind of intracellular recycling system. So as this process is a bit complicated, so I'd like to show you a CG movie that was created by Tomoko Oshima, a former undergraduate medical student. So now part of the cytoplasm is engulfed by this autophagosome. So sometimes mitochondria and other organelles are included, and now it is almost closed. So let's look at the inside. So the outer membrane is connected to the inner membrane at this rim. And now this pore is closed. So here is a very tiny neck, and this connection is clipped like this. So the closure of autophagosome is mediated by membrane fission process, not by membrane fusion. They actually, so Dr. Takahashi's group and Stenmark's group reported that escort machinery is important for the closure step of autophagosomes. So once it is closed, lysosomes, so usually more than one lysosomes fuse with a single autophagosome to degrade the inside materials. So my lab has been working on autophagy to elucidate its molecular mechanisms and the physiological functions. So to visualize autophagosomes, we have used LT3 as a marker. So these are mouse embryonic fibroblasts expressing GFP-LC3. So under normal growing conditions, there are only few autophagosomes like this. So but during starvation, the number increases to 100 or 200 per cell. So autophagy is a highly dynamic cellular process that responds to starvation. So at a higher magnification, so you can see the formation of autophagosome like this. So it takes about 5 to 10 minutes. So we have applied this method to study autophagy in whole animals like mice. So we generated GFP-LC3 transgenic mice, and we can know when and where autophagy is induced. So for example, autophagy is induced almost all tissues during starvation, after birth, and also after fertilization. And autophagosomes accumulate in some disease models, such as acute pancreatitis. Okay. The function of autophagy can be roughly divided into two groups. The first one is production of amino acids and other degradation products. So this function is important for starvation adaptation and early development, embryonic development. The second function of autophagy is, of course, intracellular clearance or quality control. So autophagy can degrade abnormal proteins and some organelles and even invading bacteria. So this function is quite important for prevention of tumorigenesis and neurodegeneration. So how these functions relate to human diseases has also been elucidated these years. So in collaboration with Dr. Naomichi Matsumoto's group, so we have identified mutations in 45 VP4 genes. That is one of the mammalian homologies of East18 in a very rare neurodegenerative disease called SENDA or BPAN. So this is a biphasic disease. So during early childhood, so they progress only non-progressive psychomotor retardation, but they can walk. However, after adulthood, this is the same patient, so they suddenly develop very severe dystonia and Parkinsonism and become bedridden like this. And we confirmed that autophagic activity is actually decreased in patient blood cells. So a defect in autophagy may contribute to pathogenesis of this disease. Okay. So now I'd like to talk about organelle degradation. So autophagy used to be considered as a non-selective bulk pathway. So, but now it is very clear that autophagy can selectively recognize some specific cargoes, such as some proteins, organelles like depolarized mitochondria, the EL, and damaged lysosome, and intracellular pathogens. So in fact, organelle degradation is one of the hottest topics in the field of autophagy research. So this is an example. So in this EM picture of a mouse embryonic fibroblast, so this autophagosome is now trying to engulf this huge mitochondria. So this is called mitophagy. And on the other side of this autophagosome, so you can see the endoplasmic reticulum. So this is called Autophagy. So however, from now, so I'd like to talk about a completely different type of organelle degradation in the lens. So that is independent of autophagy. So they are major contributors, Hideaki Morishita and Tomoya Eguchi. The lens of the eye here. So this is a very beautiful transparent tissue. And it is it consists of epithelial cells and fiber cells. And these fiber cells originally contain a lot of intracellular organelles, like the endoplasmic reticulum, nucleus, and mitochondria. So however, all of these organelles were degraded during differentiation, forming the organelle-free zone. Okay. So although this is a very dramatic process, the molecular mechanisms of this process has been largely unknown. So these are mouse embryonic lenses. So this is a lens at embryonic 70.5. This is just 2 days before birth. So you can see plenty of organelles. So green indicates the endoplasmic reticulum, red indicates nucleus. So but only just after 2 days, these organelles are degraded in the central region, forming the organelle-free zone. So cells are there, but they don't have organelles. So at first, we thought this process should be autophagy, because we were working on autophagy, but we were wrong. So when we look at a lens from ATG5 knockout neonates, so we observed a very clear organelle-free zone, even without autophagy. So it means there should be autophagy independent pathway in the lens organelle degradation. So actually, so we realized this finding way before the publication of this paper. So maybe around in 2003 or 2004. So we have been working on this project almost 20 years, or at least more than 15 years. Okay, so to reveal the mechanism behind this organelle degradation, we first established a live imaging system using zebrafish. So this is a very nice system. So this is just one example. So we generated detomate transgenic zebrafish expressing G tomato inside the nucleus, and we put the fish on the stage of microscope and performed live imaging. So these are nucleus in the lens at 50 hours post-fertilization. Then you can see that these nuclei were degraded from the center to the periphery like this. So this is very nice system. So we don't have to sacrifice these fish, so they are just indeed alive. Okay, so this is another example. So we made a transgenic line expressing EL matrix GFP and mitochondrial matrix RFP, and did time-lapse imaging from 60 hours post-fertilization. So please look at the central region. So now you see many mitochondria and the EL. So, but these organelles were degraded from the center to the periphery. So because we used matrix markers for EL and mitochondria, so this result indicates that these organelles are ruptured in the lens. So then what is the mechanism of this membrane rupture? So we performed CRISPR screening. So we selected around 60 genes that are highly expressed in the lens compared with other tissues. And we performed F0 knockout analysis using CRISPR. We injected guide RNAs into one-cell embryos. And after 3 days, we directly observed the lenses of this fish. And finally, we identified PLAT1, that is phospholipase acyltransferase, as a gene that is required for organelle degradation in the lens. So PLAT1 is actually highly expressed in the lens. Okay, so to elucidate the function of PLAT1, we generated PLAT1 knockout zebrafish. So as I told you, mitochondria and the EL are degraded between 50 to 70 hours post-fertilization, like this. So, however, in PLAT1 knockout zebrafish, mitochondrial degradation was suppressed, and also EL degradation was suppressed. So these results suggest PLAT1 is required for degradation of organelles in the lens. The PLAT1 enzyme family is highly conserved in vertebrates. And mice has 3 PLAT1 homologs, PLAT1, 3, and 5, and only PLAT3 is highly expressed in the lens. So we made PLAT3 knockout mice. This is a control experiment using heterozygous mice. So at 0.5 p 0.5. So as I showed you, EL and mitochondria were degraded in the central region. So, but in PLAT3 knockout lens, EL degradation and mitochondria degradation was suppressed, and maybe you can see intact mitochondria even in the central region. So this is completely abnormal. So the function of PLAT in the lens is conserved in vertebrates, in mice. Okay, so if we have PLAT family lipases, EL and mitochondria were ruptured in the lens, but without PLAT1, these organelles were not degraded. So next, we investigated whether PLAT family lipases are indeed present on these organelles or recruited to these organelles when they are degraded. PLAT1 has a large lipase domain and the C-terminal transmembrane domain. So this is a tail-anchored protein. So before the onset of organelle degradation, PLAT1 was diffusely present in the cytosol in lens fiber cells, even though it has a transmembrane domain. However, during organelle degradation, the PLAT accumulates some punctate structures like this. So now mitochondria are degraded and PLAT forms punctate structures. Okay, this is a highly magnified image movie of PLAT1 and mitochondria matrix marker in the central region of the lens. At first, PLAT1 was not detected on these mitochondria. However, then PLAT1 was recruited and mitochondria were degraded. So PLAT1 was recruited, mitochondria were degraded. Okay, and these are frames of this movie. So when PLAT1 was recruited, mitochondria were degraded within 1 hour. So I don't show the data here, but we confirmed that lipase activity is required, and the transmembrane domain is also required for membrane targeting of PLAT1. We also wondered about the biological significance of this PLAT-dependent organelle degradation in the lens. So as you see here, the lenses from PLAT1 knockout zebrafish and the PLAT3 knockout mice were less transparent, and the grid patterns behind these lenses were distorted. So it suggests that PLAT1 dependent organelle degradation should be important for optimal lens transparency. Okay. So this is a summary of the this part. So I'm very sorry that I skipped many of the data today, but let me just summarize. So PLAT1 family phospholipases translocate from the cytosol to organelles to degrade their membranes. So to recruit PLAT1, so we found that these organelles must be already partially damaged, but this damage is not sufficient for organelle degradation, and these organelles were degraded only after PLAT1 recruitment. So, but they are partially damaged. So currently, we don't know how this minor membrane damage is generated, but we found that this membrane damage is spatio-temporally regulated by the transcription factor HSF4. So HSF4 is activated from the center to the periphery. So I think we can explain why membrane degradation proceeds in the same order from the center to the periphery. So, but we don't know the exact what happens between HS4 activation and the membrane damage. So, but in any case, the PLAT1 is essential for complete organelle degradation in the lens. So it is well established that the degradation of intracellular organelles is mostly mediated by canonical autophagy. So, however, we found that organelles in the lens is degraded by PLAT-dependent and autophagy-independent process. So we think that this is more efficient compared with conventional autophagy. So when we need to degrade everything in the cell, because autophagy needs to envelop each piece one by one. Okay. Okay, so next I would like to talk about another story, ubiquitination of membrane phospholipids. So this work was done, mostly done by our assistant professor, Junichi Sakamaki. Okay, so this is a graph of autophagy papers appeared in PubMed. So Dr. Yoshinori Osumi published his first autophagy paper in 1992. Yeah, and next year, so this young Osumi and his master students Miki Tsukada reported 15 autophagy mutants called APG mutants. And next year, 1994, Mike Toom and Dieter Wolf's group reported another set of autophagy mutants called AUT mutants. And in 1995, Dan Klionski reported CVT mutants. So isolation of these autophagy deficient mutants led to the identification of many autophagy genes that are now called ATG genes. So I joined Dr. Osumi's lab in 1997. So as you see here, so at that time, there were only a few papers published in this autophagy field. So it was a happy time for us because we didn't have to read many papers. So what we should do was just doing experiments. And but now I'm very sorry that now more than 100 or 200 papers were published every week. So you have to read many papers. So I was very lucky, and actually I was very lucky to be able to publish my first paper about autophagy. This is about ATG protein conjugation system. So in this ATG system, ubiquitin-like ATG12 is conjugated to another ATG protein, ATG5. The C-terminal glycine of ATG12 is conjugated to a lysine residue in ATG5, and this conjugation is mediated by two other ATG proteins. Actually, the C-terminal of glycine of ATG12 is activated by E1-like enzyme ATG7 and transferred to E2-like enzyme ATG10. And finally conjugated to ATG5. And later we found another ubiquitin-like system, ATG8 system. So in this system, ATG8 is activated by the same E1 enzyme ATG7 and transferred to ATG3, and finally conjugated to a phospholipid, a phosphatidylethanolamine, not to a protein. And ATG12/5 conjugates can facilitate the final step of ATG8 conjugation. So it is like an E3 enzyme for the ATG8 system. And it was my great honor that Yoshinori Osumi used my data in his Nobel lecture in 2016. So I did this Western blotting by myself about 25 years ago to show that ATG12 is conjugated to ATG5. This is more general scheme. So it is well known that ubiquitin and the other ubiquitin-like proteins such as NED8, SMO, and ISG15 are covalently conjugated to a protein substrate, and which is catalyzed by E1, E2, and E3 enzymes. Maybe only one exception is the ATG8 system, as I told you. This is ATG8 in yeast and SE3 or GABARAPS in mammalian cells. And in this system, ATG8 is conjugated to phospholipid PE. So it occurs because the structure of the head group of PE is quite similar to that of lysine side chain. The C-terminal glycine of ubiquitin and ATG8 are conjugated to these amino groups. Then, so it raises a question. So whether a lipid conjugation is indeed specific to ATG8. So ubiquitin or ubiquitin-like proteins may also be conjugated to phospholipids. So we tested this hypothesis to do that. So we tried to collect hydrophobic ubiquitin species from yeast and mammalian cells. So organelle membranes were isolated from these cells and solubilized in a buffer containing Triton X114. Triton X114 buffer is homogeneous at 0 degree. So, but after heating to 37 degree, it separates into the aqueous and the detergent Triton X phases. And from the detergent phase, we precipitated hydrophobic ubiquitin species by immunoprecipitation. So it should contain ubiquitinated hydrophobic proteins like transmembrane proteins and ubiquitinated lipids, if any. And by immunoblotting, we detected so many hydrophobic ubiquitin species from the detergent in the detergent fraction. And there is a band that was slightly larger than the canonical endogenous monoubiquitin. And most of these signals disappeared when we deleted the C-terminal glycine of ubiquitin, so suggesting that these hydrophobic molecules are conjugated to the C-terminal glycine of ubiquitin. So if phospholipids are included in these species, so it should be they should be cleaved by phospholipase D. And in fact, this slightly larger ubiquitin band was reduced after PLD treatment, and it shifted downwards a little bit to the original position. And this band was reduced by phospholipase D, suggesting that these should be phospholipid conjugates. This data was from yeast, and we also observed the same thing for mammalian cells. So this slightly larger ubiquitin band was degraded by phospholipase D and moved to the original position. And we also observed a similar result for endogenous ubiquitin. So in this case, we used the UVA domain from ubiquitin-1. And so we mainly detected tri-ubiquitinated or di-ubiquitinated phospholipids. So because the ubiquitin-1 UVA domain has lower affinity to monoubiquitin. And although I don't show the data here, but we also found that phospholipids can be polyubiquitinated. But for simplicity, we focused on this monoubiquitinated phospholipids. Okay, so by analogy to ATG8, we hypothesized that ubiquitin must be conjugated to PE. In yeast, PE is synthesized by via these two pathways. So when we deleted major enzymes PSD1 and PSD2, the amount of this phospholipid was reduced, suggesting that the target substrate must be PE. To directly identify the substrate, we performed mass spec analysis. So we purified ubiquitinated phospholipids from yeast cells, removed their acyl chains by saponification, digested them with lysyl N-peptidase, and finally subjected them to mass spec analysis. This is a control experiment. From the aqueous fraction, we detected the C-terminal fragment of ubiquitin. So on the other hand, from the detergent Triton X140 fraction, we detected the C-terminal ubiquitin fragment modified with glycerol phosphorethanolamine, which is a head group of PE. So this data suggests that ubiquitin is indeed conjugated to PE. And we also detected C-terminal fragment modified with glycerylphosphorylserine, a head group of PS. But the amount was very small, so it should be a very minor population. Okay. Many years ago, in 1995, a paper suggested that ubiquitin is attached to membrane of baculoviruses, virus particles. The authors suggested that phospholipid is conjugated to internal serine or threonine residues in ubiquitin, and not to the C-terminal glycine. So we tried to reproduce this result. And we indeed detected hydrophobic ubiquitin species in baculovirus particles, but it disappeared when we deleted the ubiquitin C-terminal glycine. And also we directly detected the ubiquity GPE conjugated the C-terminal fragment of ubiquitin. So this data suggests phospholipids PE is conjugated to the C-terminal glycine, not to the internal serine or threonine. So our data suggests that ubiquitin is conjugated to PE in budding yeast, mammalian cells, and baculoviruses. Next, we determined the subcellular localization of ubiquitinated PE. So we separated yeast organelles by membrane flotation analysis and found that ubiquitin PE was mostly co-fractionated with the endosome marker PEP12 and some co-fractionated with the vacuolar marker 480. Indeed, we detected ubiquitin PE in purified endosomes and purified vacuoles. So when we did this experiment in mammalian cells, we detected ubiquitin PE in endosomes and lysosomes, but not in autophagosomes. And consistent with this result, the level of ubiquitin PE was increased upon inhibition of vacuolar degradation with concanalysin A. So this data suggests ubiquitin PE should be incorporated into intraluminal vesicles and degraded inside the vacuole. And we detected ubiquitin PE even under normal conditions, but the amount was increased after nitrogen starvation in yeast and mTOR suppression in HeLa cells. So although these two conditions can activate autophagy, we confirmed that this upregulation is independent of autophagy. We observed the same thing in autophagy knockout, ATG knockout cells. Okay. So next we tried to identify enzymes required for ubiquitination of PE. And first we confirmed that E1 enzyme is required. And next E2, so we screened the E2 enzymes in yeast and found that UBC4 is very important, but there was still some signal in UBC4 mutant, but if we deleted both UBC4 and its homologue UBC5, the signal was completely gone. So UBC4 and UBC5 are E2 enzymes. Next E3. So we selected three E3 candidates, TAL1, PIP1, and PIP5, So because they are known to interact with UBC4 and they are present in the endosome or vacuole. So among these three E3 enzymes, only TAL1 mutant show a reduction in ubiquitin PE. So TAL1 should be the E3 enzyme. So TAL1 was originally identified by Fulvio Reggioli. So before he came to the autophagy field, and TAL1 is a multi-spanning transmembrane ubiquitin ligase with a ring domain. So TAL1 is present in the Golgi endosomes and the vacuole, and already known to be important for the quality control of membrane proteins by promoting their degradation in the vacuole. Okay, and finally, we found ubiquitin PE is deconjugated by DOA4. So the fate of ubiquitin PE is either deconjugation by DOA4 or degradation in the vacuole. Okay, so next we try to confirm that these enzymes are sufficient using in vitro reconstitution system. So we used the TAL1 ring domain with a histag to attach liposomes to mimic endogenous multi-spanning TAL1 enzyme. And we confirmed that in the presence of all these enzymes, E1, E2, E3, and PE conjugate containing liposomes, we were able to detect ubiquitin PE like this. And if we used PS containing liposomes instead of PE liposomes, we didn't see ubiquitin PS, or very, very small amount of ubiquitin PS. So the PE should be the major target substrate. And if we use GFP ubiquitin, we were able to detect GFP ubiquitin on the surface of PE containing liposomes in the presence of these enzymes. So, but again, PS was not detected on the surface of liposomes. Okay. So now, we come to a very important question. So what is the role of ubiquitin PE? So what I have shown you is that ubiquitin PE is present in the endosomal membranes and the vacuole viruses. And it is known that intraluminal vesicle formation and vacuovirus budding require the escort machinery. And it is thought that ubiquitinated proteins on the endosomal membrane recruits the escort machinery. Then we hypothesized that ubiquitin PE in the endosomal membrane may also recruit the escort machinery and contribute to the formation of intraluminal vesicles and virus budding. So we checked intraluminal vesicle formation. So in yeast, so FM464 can label the vacuolar membranes like this. So, but NBD labeled PC is incorporated into the intraluminal vesicles and delivered to the lumen of the vacuole. So we detected NBD-PC inside the vacuole in Y-type yeast cells. And as I told you, so UBC4 and UBC5 are required for ubiquitin P formation generation. And in UBC4-5 double mutants, we didn't see NBD-PC accumulation inside the vacuole, suggesting that UBC4-5 are important for intraluminal vesicle formation. But this data does not necessarily indicate ubiquitin PE is important for intraluminal vesicle formation, because UBC4 and UBC5 are also important for protein ubiquitination. So we can't separate them. Then we used in vitro reconstitution system to check if ubiquitin PE can recruit escort components. So we again conjugated ubiquitin to PE containing a liposome like this, and we've confirmed that to these liposomes, the escort 0 components BPS27, HSE1, and the escort 1 component BPS23 were recruited. So we think that not only ubiquitinated proteins, but also ubiquitinated PE can contribute to the recruitment of escort machinery. We hope so. And finally, we checked other ubiquitin-like proteins and found that NED8 and the ISD15 have hydrophobic species that were sensitive to PLD. And the formation of this band depends on their C-terminal glycine. So this data suggests that not only ATG8 and ubiquitin, but also NED8 and ISG15 can be conjugated to phospholipids. So the conclusion of this part is the conjugation to membrane phospholipids is not specific to ATG8, but could be a more general feature of the ubiquitin family. Okay, so taking the remaining a few minutes, I'd like to quickly talk about a new reporter, Autophagy flux reporters. So they are contributors, Wheeler Im and Hayashi Yamamoto. So it is very important to measure autophagic flux in most autophagy studies. So in NIST, the most commonly used autophagy flux assay is probably GFP-based reporter processing assay, for example, using GFP-ATG8. So under normal growing conditions, GFP-ATG8 appears in its full length. So, but after autophagy induction, such as by rapamycin treatment, free GFP fragment appeared. So this is because GFP is relatively stable inside of a vacuole. So this is a very useful, very nice assay system, but it is not easy to carry out in mammalian cells. So because GFP itself is degraded inside mammalian lysosomes like this, so even if we observed degradation of GFP-AC3, we couldn't see accumulation of GFP. So if we use RFP instead of GFP, so we actually detected RFP fragment because RFP is more stable than GFP in mammalian lysosomes. However, it may be too stable, and we often detected RFP fragment even before autophagy induction due to basal autophagy flux. So the difference is often unclear. So we need a better tag for the processing assay in mammalian cells. Then we found that HALO tag is very useful for the processing assays. HALO tag is a self-labeling protein that covalently binds to its ligands. First, we tested HALO LC3. So without the ligand, HALO LC3 was degraded during starvation. But again, we couldn't detect accumulation of HALO fragments. So it suggests that a ligand-free HALO was degraded in mammalian lysosomes. However, after ligand treatment, so we could detect HALO fragment. So suggesting that ligand binding stabilizes HALO tag inside the lysosomes. So this band was not detected in 1500 knockout cells. So the production of this band completely depends on autophagy. So we can use HALO for the processing assay in mammalian cells. So using these HALO-based reporters, we can do pulse chest analysis. So, for example, the amount production of halo was very nicely detected during starvation. Its amount was increased. It's very linearly, and the results were highly reproducible. So we can use halo tag not only for LC3 but also for any selective cargos such as mitochondria and the ER. And finally, we can monitor non-selective bulk autophagy using halo GFP reporter, which is engulfed by autophagosomes only randomly. So even though it occurs at low levels, we were able to detect halo fragment during starvation in white type cells, and it was completely blocked in 5200 knockout cells. But still, we were able to detect some halo processing in even in ATG3 knockout and ATG5 knockout cells. So it suggesting the leaky phenotype of these ATG conjugation deficient cells, which were reported by many recent papers. So the conclusion is the halo tag-based processing assay is simple and reliable way to constantly monitor various forms of mammalian autophagic flags. So I highly recommend this method, and it is actually a golden standard method in my lab. So these reports are already available at Gene. So please use any experiments in your lab. Okay, thank you. I'd like to thank all the people who contribute to these works. Okay, thank you very much for your attention.
Thank you, Noboru. That was absolutely fascinating. And I think I speak in name of many scientists already. Thank you for sharing all these plasmids on AdGene. So I think they're going to be super useful. I'm just quickly going to open the Q&A box, see if there's questions have come in, because we have a few minutes left. There haven't, but if people still want to ask a question, you can still quickly type it and we'll receive it. But maybe I can go with a question I had about the first part of your talk. In the plate 1 or plate 3 system, the fact that it would always be cytosolic and then under membrane leakage or damage, it would get inserted into the mitochondrial surface. It sort of reminded me of how the PINK1 system works. I was wondering, like, did you make a comparison? Are plate 1, plate 3 in a way also processed? Are they continuously imported, or did it rather say cytosolic? So the question would be like whether there's similarities between the two systems or whether they actually function very differently.
So you're asking whether the plate 1 the lipase is the relationship between the lipase and the mitochondria leakage, maybe.
Yes. The question is whether there's similarities with how PINK1 is processed and the lipases.
That's a good point. And actually, we detected the lipase recruitment during parking-dependent mitophagy. So we found that the parking PINK1 system is very strong, and we published that the outer mitochondrial membrane was broken during PINK parking dependent mitophagy. And in that case, plate 1 phospholipase is actually recruited to mitochondria. So it may help the break of the outer mitochondrial membrane. But now we are trying to see more physiological situations whether plate phospholipase contribute to the pore formation of mitochondria under more physiological conditions. But now we have that experiment.
Okay. Some questions from the audience. There's a question from Vinay Ipan. He's asking what prevents ubiquitin of PE on autophagosome. Is it the localization or is it the E3?
Yes, That's a good point, and I think so. In this case, the E3 enzyme TAL1 is mostly present on endosomes, not on autophagosome. So I think that is the reason why autophagosomal PE is not ubiquitinated. But there is a ATC8 conjugation system, so PE is conjugated with ATC8.
I see. We also have a question from Jeremy Trone. I think he'll also raise his hand. So maybe Doro, could you help Jeremy to be unmuted?
He's already online. He can.
Okay. Jeremy, you can go.
In the NBD FM464 experiments in yeast, what happened in a TAL1 delete mutant? That's my first question.
Yes. Yeah, TAL1, yeah, we did that experiment, but the phenotype is very mild. So because we still see some ubiquitin PE in even TAL1 delta mutant, but it was not detected in ubiquitin UBC45 delta mutants. So then we chose to see UBC45 delta mutant. But the phenotype is much more mild in TAL1 mutant.
So do you think the PIB or one of the other E3s might act as a backup? Is that?
Yes, I think so.
The other question had to do with the fact that in sphingolipid biosynthesis, of course, the long chain base precursor also has a free amino group before it's amidated with a fatty acid. Fatty acid. So especially if you block ceramide synthesis, can you see ubiquitin attached to the long chain base as well as to PE and PS?
Yeah, thank you for asking. So that is another very interesting point, and actually we are working very hard on other lipids molecule, and the sphingomyelin may be another good target. And so it is also known that ubiquitin can be conjugated to hydroxy groups, not only to amine groups. So I think there are many more non-protein substrates, but now we are working on that. Thank you.
And then I think we have one last question. It's a very short one from Elliot Mock. He's asking, what's the mammalian homologue of the ubiquitinase DOA4, where you've tried knocking it out?
Yeah, thank you. That is another good question. So now we are trying to identify mammalian counterparts, but so far we couldn't find any in one. So TAL1, that direct homologue of TAL1 is not present in mammalian cells. I think some endosome localizing E3 enzymes may have some similar function, but anyway, we haven't identified the enzyme. Maybe redundant.
I see. So, well, then once again, a big thank you, Noboru, for this fantastic webinar. And I would like to invite everyone for our next speaker on the 27th of February. It's going to be Ai Yamamoto from Colombia. I hope to see many of you there again. And then that's it for today. Bye.
Thank you.
Thank you very much.